So we are in the process of grading your exams. We finally were able to load everything on Gradescope, so we'll be getting into that pretty quickly. Your multiple choice parts are going to be scanned today, and we will immediately upload that to Canvas, so you will have those results later today. If you haven't done this, you will get access to that through the scanning services on the Canvas menu of our course page. And that will show you your results with also the key. So we'll tell you which questions you got right and which ones didn't. All right? Any questions? Today we're going to continue our conversation on nuclear transport. If anybody forgot their handouts, I have a few left. So just let me know, and we will pass them around. I'm going to ask you to pass them around. Thank you. So, and I know this is all stuff we talked about before the exam, so maybe we've forgotten some about it. So let's try to recall where we were at. So we were talking about considering how proteins are going to be transported from their site of synthesis in the cytoplasm to then be targeted or transported towards the nucleus. Proteins also have to exit the nucleus, so proteins that were previously functioning in the nucleus may need to be transported back to the cytoplasm. So there has to be a mechanism for that as well. In this case, we're talking about all soluble proteins. So we're not considering, there are special cases of intramembrane proteins that are found in the nuclear envelope. We're not going to consider those. We're only going to be considering soluble proteins. Proteins that are synthesized by cytosolic ribosomes, that are floating completely in the water environment of the cytosol and need to be transported to the nucleus. All transport has to occur through the amazing structures of the nuclear pore complexes, places at which the outer membrane and the inner membrane are fused with each other, leaving an open hole that is gated by a very large collection of proteins that form a complex, a nuclear pore complex. Those are the proteins that are going to determine what things can get in and out and which cannot. We talk about how a subset of nucleoporins, proteins that form the nuclear pore complex, form a hydrogel, so they are disordered but form a gelatinous structure that has an open hole in the middle. So anything that is able to fit the open hole will be able to pass in either direction simply by diffusion. That will include ions, all nucleotides, other small molecules, and small proteins all the way up to around 50 kilodaltons. It all depends on shape and whether they can actually fit through that hole. Right? So anything that is larger than 50 kilodaltons will be rejected by this gelatinous mass unless it has a proper signal to travel in one direction or the other. So to go into the nucleus, we have a nuclear localization signal, or NLS. To get out of the nucleus, we have a nuclear export signal, or NES. All right? Since we have signals, we talk about how the nuclear localization signal, the only one you need to know is the one that is 5 positive charged amino acids on the surface of a protein, lysine or arginine mostly. And because these are amino acid signals, there has to be a protein that acts as receptor. So we have a variety of signals. Proteins can have a variety of signals. The only one you need to know is the basic NLS. But there are a variety of ones, and each of those different signals will be recognized by unique different receptors. Receptors that are able to bind by non-covalent bonds to those amino acids in the signal. So some of them may be for import, some of them may be for export. Right. So we're going to have nuclear import proteins or nuclear import receptors, and we're going to have nuclear export receptors. There are different proteins from each other. Signals are separate, receptors are separate. And we then went into the experimental evidence that shows that nuclear import receptors, I didn't show you this for export receptors, but I show you this data for a nuclear import receptor, has the ability of interacting with the proteins of that hydrogel, bind tightly to them, so it has a high affinity for those proteins in the hydrogel, and is completely able to enter the hydrogel. It's able to penetrate the hydrogel. And in doing so, is able to carry with it any protein that it has bound to. Any protein that has the correct signal, grabs it by the signal, and as it moves into the hydrogel, it can bring that other protein as a passenger. Again, I need to reiterate this. This is going to be through the hydrogel here for things that are not able to fit in the hole. All right? So let's consider if we had somehow made up a nuclear pore that was completely 100% filled with the hydrogel without an open hole, even small proteins would not be able to get through because it's a very solid mass. All right? Small proteins can get through only because there's an open hole. All right? And anything bigger then has to squeeze through the hydrogel, and that is a property, unique property of the import receptors. How do we know that a protein that is a passenger protein with a signal can actually get through the hydrogel? Because we did the last experiment showing that a cargo protein that has the NLS sequence will enter the gel, will not enter the gel if it is by itself. But if you incubate it with the import receptor that is not visible in this image, then the protein, the passenger protein, the cargo protein can enter the gel. It is being dragged into the gel by the import receptor. All right? So this is where we left last time. Anybody has any questions on this? Any wonderings, amusing thoughts? Yeah. A little louder, please. So the receptors are also soluble proteins. So some of them will be found in the nucleus because they got there. Some of them will be found in the cytoplasm. All right, all good? Great. So now we need to consider how is it that the receptors actually work. So we understand that they have amino acid sequences somewhere in their binding pocket that are going to be able to interact with the amino acids of the signal of the NLS. So say if you have negative amino acids in your binding pocket, they will be able to interact by charge interaction with those 5 positive amino acids of the signal. That's easy to understand. The problem, though, is that this interaction has to be regulated. It has to occur in one location and not occur in the other. The receptor has to bind in one location and release in the other. And we did that for the KIL receptor. We did that for the mannose-6-phosphate receptor. We did that for LDL receptor. All those 3 receptors can interact with their cargo, with their specific proteins that they bind to, and release in another location by a change in pH. So from more or less acidic one way or another. Here we don't have a pH difference. pH is the same in both sides. Salt is the same. Ion concentration is the same in both sides. Nucleotide is the same in both sides. Everything is the same in both sides. So how does it work? How do you turn it on in one side and turn it off on the other? The key for this is a small protein called RAN. It's a tiny little protein known as RAN. What is RAN? We're not talking about this famous movie by Akira Kurosawa. How many of you have seen it? Finally, one person, thank you. Last year I had nobody. How do you like it?
I liked it.
I think it's an awesome movie. So it's a classic movie. It's the story of King Lear, Shakespeare's King Lear, but told in the samurai tradition. No CGI, but they have amazing battle scenes with thousands and thousands of extras and horses. It's gorgeous. Anyways, so highly recommend the extra points now. So RAN. RAN stands for RAS protein, RAS-related nuclear protein. Means nothing, perhaps to you. This is another small GTP binding protein. Just like we already talked about SAR1 and ARF1 as being examples of small GTP binding proteins, and therefore proteins that are able to bind GTP. When they bind GTP, they are in the activated state. When they hydrolyze that GTP, they are inactive proteins. So this is a protein that is found in all the eukaryotic cells, very abundant in all cells. So that is RAN. So RAN exists in two states. It exists in a GTP-bound state, so where it has nice GTP bound on it. And it also exists in the GDP-bound state, where it has hydrolyzed the GDP. And remember that that transition between GTP-bound to GDP-bound was regulated also by two accessory proteins. There are the GAP proteins that accelerate the hydrolysis stage, and there are the GEF proteins, or nucleotide exchange factors, that promote the binding of a new GTP. So we have here the same thing as we totally parallel to what we did with SAR1. So we have, but the proteins are specific for RAN. So the GAP for RAN and the ARF and the, too many three-syllable things. The GAP for RAN and the GEF for RAN are unique for RAN. They will not interact with SAR1 or any of the other small GTP binding proteins. So because of that, we're going to just call them RAN GAP and RAN GEF. All right? So RAN GEF, now this is the key. Just as the key for why you bud COP2 vesicles only from the endoplasmic reticulum. The reason was that the GEF for SAR1 exists only in the membrane of the endoplasmic reticulum, and therefore you activate SAR1 only in the membrane of the endoplasmic reticulum. So the same thing applies here. RAN-GEF is a protein that itself is localized inside the nucleus. RAN-GAP, on the other hand, is a protein that exists on the cytosolic side of the nucleus. So the difference in localization of the GEF and the GAP is going to set up a different activity of RAN in those two locations. RAN, if RAN is inside the nucleus, it will be able to bump into RAN-GAP. And the effect of that is that GEF will tell it, grab a new GTP. So it will grab a new GTP, will be in the GTP-bound state. Now it can do something. It can interact with something or someone. That something or someone will be crucial for this to work. And we'll go into that in a minute. If RAN happens to make its way to the cytosol, on the other hand, it will interact with RAN-GAP, which will kick it so that it hydrolyzes the GTP. And now it will be in the inactive state, and it cannot interact with anybody. All right? So we have set up a, I don't like to call it a gradient, but it's often described as such, a gradient of RAN activity. RAN is active inside the nucleus, and RAN is inactive in the cytosolic side. Simply by localizing those two accessory proteins in those two different locations. Questions?
So having the gap in the cytosol